Good day, this is Professor Will, CBMD PhD. Today's Monday, October 19th, 2020. It is 5:12 and 37 seconds a.m. Eastern Standard Time, and this is a continuation of my microbiological research. So let's hop to it here. Concerning the phylogeny-based description of higher taxa, the domain Archaea has received considerable attention primarily because of research activities in the laboratories. When higher taxa were proposed for the second archaeal kingdom, the Crina archaea, only a few species had been described. A similar situation is encountered among the deeply branching lineages of domain bacteria. Encouraged by the ease at which higher taxa were described for archaeal lineages, orders and families were almost simultaneously described with the recognition of a new genus or cluster of neighboring genera. Examples are order of Aquaficales, embracing the family of Aquaficae, and the genera of Aquafix, Calderobacterium, Hydrogenobacter, and Thermocrinus, as well as the order of Thermodogales, embracing the family of Thermodagaceae, and several genera, Thermodoga, Geodoga, and Petrodoga. These examples demonstrate that the description of higher taxon for a phylogenetic lineage is facilitated by the small number of organisms and by the isolated position of the lineages. The situation is different in the four main bacterial lineages, which show a complex phylogenetic structure. The gram-positive bacteria division, Firmicutes, the class Proteobacteria, the Bacteroideaceae, Cytophagales line of descent, and the Cyanobacteria phylum. Within the Cyanobacteria lineage, higher taxa have been described on the basis of morphology and within the prochlorophytals of pigment composition. Analysis of 16S rDNA clearly demonstrates that the presence of affiliation of genera into the orders of Crucocales, Nosticales, Oscillatoriales, Procapsules, and Stigonomatales is not always supported from the phylogenetic point of view. One of the most unexpected relationships that emerged from the analysis of rRNA, rDNA, that certain genes coding for proteins was the specific groupings of the Bacteroids and Cytophageal Flavobacterium lines of descent. No higher taxon has been proposed as yet for the lineage comprising a phylogenetic assemblage of genera, but within the exception of phylogenetically coherent family of Spingobacteriaceae, organisms are related to the traditional higher taxa Cytophagales and Bacteroideaceae. The gram-positive bacteria constitute a division of Firmicutes, which with the exception of members of the Deinococcus appears to form a phylogenetically coherent taxon. The common ancestry of the two major sublines, however, one embracing the actinomycetes, the other containing the Clostridia, bacilli, and their relatives, has not been convincingly demonstrated by the rDNA analysis. If for the time being it is assumed that these organisms indeed share a common ancestry, then the deep separation between organisms with a DNA-based composition of less than about 30 mol% G plus C, the Clostridium bacillus lineage, as those with a higher G plus C content, the actinobacteria may facilitate resolution of their hierarchical structure. A hierarchical classification system has been described for the actinomycetes and their relatives. The rationale for doing so is based on the fact that nearly all type strains of validly described species were characterized phylogenetically and that genera constituted phylogenetically and phonetically coherent taxa. The decision to classify phylogenetically neighboring genera and the families neighboring families into suborders and to continue up with the level of class actinomycetes was done irrespective of the phonetic properties on which higher classification of these genera was based in the past. Rather than focusing on the characteristics such as morphological, physiological, and chemotaxonomical traits, which except the presence of mycolic acids have no or only restricted phylogenetic meaning above the genus level ranks, were defined on the basis of emerging phylogenetic clusters in the presence of taxon-specific rDNA signature nucleotides. As a consequence, the actinomycete proper has been classified into 5 subclasses, 6 orders, 10 suborders, and 35 families. Each taxon is characterized by a set of 16S rDNA signature nucleotides, which were present in all or at least the vast majority of members of a taxon at the time of its description. This way of circumscribing taxa is similar to the traditional phenotype-based classification in that new members may have properties that differ to some extent from those of other members of the taxon. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and computer science. I hope you enjoyed listening. Until next time, have a great day.